Welcome to the last, let's say, the steering committee of our HUTER project. In the first place, where I would like to welcome you, and also I would like to introduce you the managing director of INCLIVA.
Okay, good morning. Welcome to INCLIVA. It's a real pleasure for us to have you here. This has been one of the most important projects in the history of INCLIVA, short history of INCLIVA, but we was born in the year 2000 as the first research institute in a hospital, in a public hospital in the Valencia community. And at this moment we manage, boost and promote the biomedical research of the University Clinic Hospital of Valencia here.
Well, first of all, it has been a big landmark, the fact that we obtained a European project as coordination here in INCLIVA. So this has been a really important issue for us. And second, to be able to coordinate these 5 different groups working in a specific pathology, in a specific organ, it has been challenging, but also it has been amazing because it gives us the possibility to interact with them, to learn from them, and also to teach them. So collaboration is everything in science. So the fact that we have been able to do this from here, from Valencia, it has been really, really important.
Yes, so it's important to look at molecular information at different levels. And by integrating different methods, you can also take advantage of the pros and cons with each method. Each method has its own advantages and different disadvantages and advantages and different type of noise. And it's especially important to note that the proteins are the molecules that carry out the functions in the cells, and proteins are also the targets of most pharmaceutical drugs.
So, as we know, the DNA in every cell is exactly the same. So the way this DNA is utilized is different to give cell specificity, and this is down to epigenetics. So epigenetics is essentially the marking of the DNA or the associated proteins to allow for differential gene transcription. And we know that this process goes wrong in certain diseases, and my lab's very much interested in those associated with reproductive outcomes.
So actually had a huge impact because at the beginning the project was relying on for samples from biopsies, but all of this got interrupted all of a sudden because of COVID, right? So at the end we had to very quickly change our strategy, and we were lucky enough that there were some historical samples stored. Of course the conditions are different, we have to optimize our protocols towards these new conditions of the samples, so I think it was a very challenging moment. But so far I think we've managed to be successful and accomplish the numbers that we suggested.
With this HUTER project, we are going to produce the map at the single cell level of the human uterus, an organ that is crucial for pregnancy, to understand how pregnancy and how the continuation of the species occurs. So by having the atlas, now we will go to the functionality. So next step is going to understand how each cell of the uterus, 6 different cell types in the endometrium and 14 in the myometrium works. So first is anatomy, and single cell is the next step in anatomy, and then will be to understand how they function together, how they relate to each other, and how to understand pathologies, uterine pathologies that still today we have no answers, such as endometriosis, such as preeclampsia, such as recurrent implantation failure, recurrent abortion, many unknown pathologies that we hope to understand with these new approaches.
So for the past 2 years we've been optimizing and using novel single-cell methylation techniques. So we've greatly improved the coverage that we can obtain from single cells for DNA methylation. And this has revealed that we're seeing certain signatures in different cell types. So what we'd really love to do now is move these signatures and look for differential DNA methylation and indeed additional epigenetic marks in different disease states at single cell resolution. So we would like to focus on preeclampsia, fibroids, endometrial cancers. These will be the follow-ups from the HUTER project.
We have been using both regular immunohistochemistry, where we stain one protein at a time using antibodies, and we have also set up a multiplex method where we're using it where we're using immunofluorescence-based methods, looking at several proteins at the same time. And using this method, you can then look at overlap between well-known and unknown proteins and look at interactions.
Well, in the HUTER project, there are a lot of data generated using cell sequencing technologies, and these technologies generate a vast amount of data. So all this data needs to be stored and processed, and the HUTER platform has a cloud architecture design which allows to use the powerful resources of the cloud and do all the experiments and analysis with higher performance than in traditional computers or servers.
So I think we are providing the reproductive community with a unique tool. So we are mapping the uterus at an unprecedented scale, and this is going to help us to try to locate all the genetic associations or the mutations associated with diseases and all this impairment, and try to start to understand why some of those diseases affect some specific woman, specific moment, and then try to start to suggest how we can develop therapies to try to help them.
We share with several companies out of the HUTER project the potential of the HUTER platform in other sectors, and all people involved in these conversations highlighted the potential of the HUTER platform in other contexts, for example in oncology, microbiology, anatomy pathology, and also in the clinical trials industry.